Hello, everyone. Uh, Welcome to today's webinar. I will give a introduction to our DNBelab C Series single-cell RNA sequencing today. Here show you the contents of my reports. I will begin with a brief introduction to the background and development of single-cell sequencing. Then I will introduce you our BGI Genomics single-cell sequencing services, the DNBelab C Series single-cell RNA sequencing. Then is the introduction of our product advantages and applications. The first part is the background of single-cell sequencing. When we talk about RNA-seq, we usually refer to the bulk RNA-seq, which obtains wait a minute. Okay. Which obtains the average information of genes at the tissue level. In contrast, single-cell RNA-seq provides gene expression information at the cellular level. When it comes to the spatial sequencing, it can help us obtain location information. So the traditional methods analyze the large numbers of cells together, and it will gain the average result obtained. It may mask significant differences between individual cells. However, even in seemingly uniform populations, cells can vary very greatly in gene expression, protein levels, and behavior. So single-cell analysis overcomes these limitations by examining each cell individually. It can reveal subtle variations in genes within different cells and thereby uncovering biological meaningful heterogeneity. When we conduct a single-cell sequencing, we need to understand the cells in a multi-dimensional ways, like the quantitative, spatial, time, and direction. When it's at the qualitative level, it means we need the diversity of cell types generated through the regulation of gene expression. The qualitative level means we need to get the count, the proportion, and the density of each cell type. The spatial level means the spatial arrangement of each cell type and their interactions within cellular ecosystem. The temporal level means the specific timing of changes in cell types and states. And last, the direction level means the potential transformational paths each cell type may undergo. So understanding these multi-dimensions of cellular diversity and organization is very crucial for comprehending the connections between the genome and the specific biology processes, as well as the regulatory mechanisms involved. Single-cell technology is revolutionizing various fields by providing detailed insights into cellular functions and interactions. So it's pivotal in oncology for developing targeted cancer therapies, enhancing assisted reproduction techniques, and in developmental biology to understand cell differentiation and embryonic development. Additionally, it plays a crucial role in spatial omics because it can be used to map gene expression across tissues and integrating multi-omics data for comprehensive biological analysis. So single-cell analysis offer a very powerful tool for advancing research and application across multi-research areas. The study of cell biological can be traced back to the invention of the microscope. And in 1990, researchers demonstrated the feasibility of single-cell transcriptomes. In 2009, single-cell sequencing was introduced for the first time. From that point on, single-cell sequencing became a very powerful tool because it can allows researchers to identify new cell types and investigate the mechanisms of diseases as a single-cell resolution. By 2018, a commercial platform had emerged. In 2018, BGI Genomics also launched our own single-cell sorting platform. It is the DNBelab C Series, and it is the newest machine. We call it 10.4. Our single-cell sequencing is based on this machine. So the next part, I will introduce you the workflow of our DNBelab C Series single-cell RNA sequencing. Here show you the overview of the workflow. In total, it can be divided into these steps. Firstly, we need to get a single cell or single nuclear suspension. Then we will use the droplet generator machine 10.4 to perform cell sorting and barcoding. After droplet breakage, molecular barcoding and amplification, we will have as a cDNA. This cDNA will be used for the library construction, and then is the sequencing and the data processing. The strategy of DNBelab C Series single-cell RNA sequencing is summarized in this table. This single-cell RNA-seq is based on the microfluidics. Here should also show you two very important indicators of a single-cell RNA sequencing. The first is the chemical cell count, and the median is the gene count per cell. The chemical cell count of our platform is between 8,000 to 10,000, and the median gene count per cell is between 1,000 to 3,000. Then we recommend you sequence on our DNB-seq platform and with 6 million row reads per sample. A single-cell sequencing is a very high cost project. So we need a strict standard during the QC process. It can help us obtain high quality data and save many costs like time and even some money. The sample preparation procedure is the decisive step of the experiment. This is an overview of sample preparation. Our services can start both from the tissue dissociation, nuclear extraction, and also the single-cell suspension. When obtaining cell suspension, we need to check for the debris. The cell viability need to be about 85%, and the cell number should be at least 100,000. Obtaining high quality cell suspension is very critical because dying cells are easily lyzed to release RNA. The release of cell-free RNA will cause background noise in the detection and affect the quality of single cell data. The next part is cell sorting and barcoding. We will use the high quality single cell suspension to do sorting and barcoding. The sorting is based on the microfluidics. And then the cell nucleus, cell beads, and the regions will flow through the channels. And then they will be ramped into the oil fluidics. So the reverse transcription will take in the droplets, and when we harvest the cDNA, we will use the cDNA to construct the library. Here show you the library construction. The cell barcode is used to label cells. The polity is used to capture mRNAs, and the UMI is used to label mRNAs. The sequencing is performed on the DNB platform. The sequencing strategy is paired and 100. It will capture about 8,000 to 10,000 cells and generating 6 million row reads. When sequencing finished, we need to perform bioinformatics analysis and visualization. The accompanying software package, DNBC4 tools, will provide fundamental analysis, including the data quality control and cell clustering. You can also use our interactive platform, .tom, to get standard bioinformatics analysis and even more advanced analysis. The standard analysis include data quality control, single cell gene quantification, cell clustering, cell type annotation, and cell function analysis. The advanced analysis include differential analysis of multiple samples, cell communication, and RNA velocity, pseudotime, and CNV analysis. Our TOM platform is an intuitive and advanced visualization system for efficient analysis and discovery. On this platform, it can enable the interaction of cells and genes data results, as well as the dynamic and static analysis of interactive mining of single cell data. It will speed up the publication process. You can also analyze your data and create your desired figures by just click on this platform. You don't need too many complex program languages like Python, R, and so on. And you also don't need to ask for the favor from your bioinformatician friend. And also you don't need any other software download. So it's very easy to use and very fast to visualization. You can perform all the analysis I mentioned before with just a few clicks on this website. If you want to simplify and customize your analysis, you need to try our TOM platform, and here show you the website you can visit. To evaluate the effectiveness of this single cell sorting platform, we also do some experiment. Here show you the sensitivity experiment. This experiment is generally use two types of cell lines with node ratios, and it was loaded on the machine I mentioned before. As we can see on the right section, trans types of cell sorting can be achieved, which is basically consistent with the preset ratio. So it further reflects the high sensitivity of single cell sorting on this platform. Our platform also have a high technical reproducibility. To evaluate the technical replication case, one PBMC samples was divided into 4, and the same batch was loaded on the 4 channels of the TEM4 machine. The cell profiles and the cell types obtained show very high consistency. Also, we have compared our data with some published data. The DNBelab C platform can efficiently sort the human PBMC samples and sequence the transcriptome library. The result single cell data were clustered using UMAP dimensionality reduction, and the results were consistent with the human PBMC cell classification and annotation result published in the Nature Biotechnology in 2020. Here show you some actual data of our platform. It also represents some typical samples, including human PBMC samples. Its capture efficiency is greater than 15%, and gene detection is about 2,500. Also, there is some human clear samples of mouse lung cancer cell samples and mouse brain nuclear samples. The capture rates are also about 50%, and gene detection is about 2,000 to 3,000. For complex samples such as OCT embedded samples commonly used in spatial temporary technology, single cell data is also available. The third part is about our product advantages. BGI Genomics has started to provide single-cell sequencing services since 2018, and we possess a rich project experience. We have our own single-cell sorting and sequencing platform. Also, Tom, I mentioned before, can also help you with data mining and accelerate your publication. We can provide you with multiple omics and multi-platform. Our end-to-end service also available. It can give you, it can help you from your sample to the data mining. Our DNBelab C Series also have helped many publications. To 2025, DNBelab C Series has facilitated the publication of nearly 300 articles, and the average impact factor is about 12, including 130 articles with an impact factor of 10 or even above. So finally, I want to introduce the applications of our single-cell RNA sequencing. We can use single-cell sequencing data for many research. Based on the research objectives, single-cell research can be divided into the following two types. We can clarify the cell type composition and the characteristics of different cell types. This is the so-called cell antlers. We can also conduct the comparative study of different states, like healthy or pathological states. Then we can describe the dynamic change process and find the cause of these changes. This idea can be used in many research areas, such as the embryonic development, tissue differentiation, cell reprogramming, tumor regenesis, immune stress, aging, and regeneration. When based on the project size and whether using multi-omics, single-cell research can also be divided as this slide. As research depends, single-cell research has gradually entered the scale-up stage. In addition to the antler research, a single-cell EQTL and a single-cell large cohort research is increasing day by day. With the development of spatial sequencing, more and more researchers also use single-cell RNA-seq to help with the annotation in spatial sequencing. A single cell can also be combined with other omics like transcript omics, genomics, protein omics, and so on. In the next part, I will show you some publication cases to help you learn more about the single-cell RNA-seq. The first is the application of C Series single-cell RNA sequencing technology. This article was published on Nature in 2022. This article explains the technical details and methods of our DNBelab C Series single-cell RNA sequencing. It based on the single-cell library construction and the sequencing platform independently developed by BGI Genomics. And the research team conducted single-cell RNA-seq analysis on about 1.40 million cells from 45 male tissues and organs. These cells are divided into 113 major cell types and 463 cell subtypes. Then this data was is used to construct the non-human primer single-cell antlers for the first time in the world. Also, this data can be available on the interactive website NHPCA. And this data can be used in the areas like species evolution, human diseases, and even drug evaluation and screen. The second study is about the engine, also the Alzheimer's disease. This study combined the DNBelab C Series single nuclear RNA-seq with the stereo-seq, and it includes 16 cases of human hippocampal tissues, including the healthy and pathological tissues. This article reviews the spatial distribution characteristics of Alzheimer's pathological changes in different sub-regions of the hippocampus and the cell type specific responses. It also reviews the molecular pathway and gives some clues to the new diagnostic targets. The third one is about the application in metabolic disorders. It also combines the single-cell RNA sequencing with the stereo-seq, and the machine is 16 to 18 years macaque hippocampus. It says 3 controls, 3 obesity samples, and 2 diabetes samples. In total, it's capture nearly 500,000 cells. It constructs a spatial transcriptomic analysis of the primary hypothalamus under the obesity and the diabetes. Also, it reviews the spatial molecular marker changes in some cell types of obesity and diabetes in different brain regions. Finally, it finds the INF-PVN pathway plays a very important role in maintaining energy balance. The fourth one is an application in the cancer area. Researchers combine the single-cell RNA sequencing with many other omics technologies like spatial transcriptome, the ATAC-seq, and CAR-seq. This article systematically illustrates the spatial interaction mechanisms between the hepatocellular carcinoma progenitor cells and the macrophages. The final one is about the usage of single-cell EQTL. Through large-scale single-cell RNA-seq and immune reptile sequencing, this study combined the single-cell data of 625 PBMC samples. The study successfully built the first single-cell EQTL map for immune cells in Asia population. The analysis is very important because it provides an invaluable resource for understanding population-specific immune biology and lays a solid foundation for developing more precise diagnostic and therapies for Asian population. Today, Okay, today I introduce the development of single-cell sequencing, the workflow of our DNBelab C Series single-cell RNA sequencing, and also the application cases of some single-cell RNA sequencing. Hope you gain some useful information from this. That's all. Thank you.